That makes sense. She's got me in it, so she's down. You're not putting in thing. But don't put her milk down the drain. What does that have to do with the milk and check? Chair. Oh. Good check. It still helps the milk check though, so it's cows.
Okay, I suppose we should get started. So lecture 13, we're in week 7. So thank you for helping with candidate review. Excuse me. I owe you 60 minutes a lecture. By my calculations, we'll try to do an extra 5 minutes per lecture where it makes sense. Depending on where the appropriate stop is. Review sessions. I handed you 5. I got to get it up on Brightspace. I'll try to do that after class. I've got almost 6, almost done. Get that one. We'll start staggering them and try to get caught up before the next exam and hopefully have a little bit of space between the assignments getting done so I can turn them back so you can get ready for the exam. So letters, let's see if we remember from the other side of break. X is the Z line or disc, the A band, the I band, or the M line. So we're looking at D, that's the M line, the center that holds the protein disc there that kind of holds all the thick filaments in the right orientation. What's this? So we're looking at the A band. How do we define the A band? Does anybody remember? The A band runs from this end of the thick filament to this end of the thick filament, so that never changes in width. What's this? So we're looking at the I band. That runs from what to what? The end of the thick filament in this sarcomere, the end of the thick filament in this sarcomere. That will change in dimensions as the sarcomere contracts. So this is what? Last one left, it's the Z line or disc that runs the entire, in our skeletal muscle, the entire myofibril through there. So what are these? The thick filament is also called the what? Myosin complex. What are these? Also called the thin filaments. The actin helix. So we're looking at the thin filaments, actin molecules formed together, linked together. You've got 2 chains running around each other. They're going to form the thin filaments. Then we're going to add troponin and tropomyosin to those. the myosin filaments. You have the long chain here, and then you have the head on the end. They come together and then arrange themselves so those heads work their way around the thick filaments. So they're going to climb up inside of these actin tubes that are formed around each of the thick filaments. So we have our myofibrils, our different sections here. Sarcomere starts here, goes to here. So we ended up talking about our sarcomeres. Why can they only contract down to 60% of where they were when they were in the relaxed state? They run into things. The thin filaments run into each other. The thick filaments run into the Z disc. That's as far as they can go. And why can't we go too long? You're going to start pulling things out, and getting things back into orientation isn't going to work anymore. I assume that's what happens on a molecular level. On that microscopic level, when we pull a muscle, we've messed up the orientation of our thick and thin filaments, and they don't interact the way they should. So how does a sarcomere contract moving forward? So you have all the different sections. How does this all come together? So one of the things that we haven't, or maybe we did, but this is review, is we need tropomyosin, this long black line, and troponin, this little kind of pill-shaped thing. They need to line up on top of our actin helix. So you've got the black lines, tropomyosin, strings of those going around up and down, and then you've got the troponin at different intervals attached to that tropomyosin. Did we talk about what the purpose of those two proteins are? My brain says no, but I wanted to double check. Okay, so when we're looking at our contraction, the tropomyosin and the troponin are going to cover up the binding sites on the actin helix. So the myosin complex, the head on the thick filament, needs a place to interact with. It needs a place to reach out to and connect with. For the moment, at least in this configuration here, the tropomyosin and troponin are covering up those binding sites. So that means what? There's no cross binding, there's no interaction. The sarcomere remains in the relaxed state. So when we think back to our composition of muscle tissue, why do we think that water might be 75 to 80% of muscle tissue? In that sarcomere, it's going to act as a lubricant. So you have those hydrophobic proteins, and the water is going to slide back and forth. So what does that water do? It's going to act as a lubricant for movement as we go through. So having that extra water there, maybe a little extra water compared to some other tissues, helps lubricate, helps things move. So how does a muscle contract? So on a macro level, we start with a signal from the central nervous system. Depending on what we're dealing with, depending on what we're reacting to, that nerve impulse is either going to start in the brain, where we have a conscious or unconscious thought, sends a signal down to the muscle fiber. Or we can have something that starts in the spinal cord. These are called reflexes. We have a lot of those happening at any one time. We'll get to those when we talk about nerve impulses. So you have that signal. It's going to come down the nerve, and it's going to end at the nerve is going to end at the sarcolemma. So this is a diagram of that happening. You have the nerve here coming down. It's got multiple buttons at the end that are going to be associated with one muscle fiber. So muscle fiber here, and then you got all the circles in here. These are the myofibrils. So we're probably looking at a skeletal muscle cell. Nucleus on the outside. We've got blood coming in. We have the button coming down here, forming the myoneural junction. We have an electrical signal coming down. That's going to release the synaptic vesicles. They're going to release acetylcholine into this synaptic cleft, the space between the nerve and the muscle cell. So that's a chemical is released. It crosses the gap on the other side. If there's enough of that acetylcholine, it will start another electrical signal that tells the muscle to contract. So the stronger the signal coming down, the more likely the signal coming across is going to tell that skeletal muscle to contract. So we end at the sarcolemma. We have that signal going across. The impulse is transferred to the muscle at that myoneural junction. Every muscle cell, particularly skeletal muscle cell, has a nerve associated with it. 1-to-1 ratio. So this gives us the name here. These vesicles, when the electrical impulse comes down, these vesicles fuse with the end of the nerve. They release the acetylcholine, the little red dots, stars or whatever they are, in the synaptic space. And then we have the receptors on the muscle cell that may or may not be excited depending on how much acetylcholine is released into that space between the two. So nerve meets the muscle cell, and we have one nerve for certainly every skeletal muscle. So if we have a 1-to-1 ratio, and if I told you that if the number of muscle cells or muscle fibers that you have is set at birth well before that, why does that make sense given what I just told you? So in order to be wired to move, I can't be changing the number of cells. So it's got to be set. So we have a nerve coming down to one cell. Now maybe there's groups of cells that move together, but you've got a nerve coming down to one cell. I can't be increasing and decreasing the number of muscle fibers, the muscle cells I have, and still make that system work. So that wiring is all set up before you're born. And what the difference is how many myofibrils are in that cell. So training is going to increase the number of myofibrils in the cell, make the individual muscle cells stronger, but it's not going to change the number of muscle cells. So we have what's called hypertrophy, each of the cells getting bigger during training, not hyperplasia, not more cells being formed. So because of that wiring system, the way the nerves are set up and how we signal those muscle cells, we have to have that relationship in advance. So as we mentioned already, acetylcholine is the transmitter. Once that goes across the gap there, it excites the receptors on the sarcolemma, the plasma membrane of the cell. They're going to stimulate the release of calcium from the T tubules. So the more signals we have coming down, the more acetylcholine, the more calcium will be released into the T tubules, and they surround the myofibrils, and the calcium will diffuse in and start the contractions. So if we go back to milk fever, if we've got no calcium there, I can't start a contraction. So we need that contraction there. So muscle contraction is a positive feedback system. The more impulses that come down that nerve, the more contraction will occur. The stronger, the longer, the more myofibrils will be activated, the stronger that contraction becomes. So the calcium is going to filter through in the system and it's going to go to the actin helix. So in order to end a contraction, we need something in that space there that's going between the nerve and the muscle that's going to break that acetylcholine and stop the stimulus on the other side. So choline esterase is going to break down that acetylcholine. If you break it in half, it's not going to work anymore. So the parts are reabsorbed by the nerve and the nerve makes new ones again. So what happens if that enzyme is not there? Does anybody know? It will continue to contract. Does anybody know what that's called? One thing it's called is called tetanus. So the tetanus bacteria will interfere with the breakdown of that acetylcholine and you will continue to contract. So on the battlefield a long time ago, we have muscles continuing to contract. So why do we get that shape? Generally, the muscles on the dorsal side are stronger than the muscles on the ventral side. So we get that bow up there, we get everything tightening up, and eventually what happens is you die because your diaphragm, the muscle that moves your lungs or fills your lungs up, contracts, and we keep the lungs in one shape and we can't get air in and out anymore and we suffocate. It also probably messes up the heart, so it's always in a contracted state. So it's not a good way to go if you're thinking about it. So get your tetanus shots. As a friend of mine works in an ER. Anybody who's ever had a tetanus shot has not died in this country of tetanus. It's good to get your boosters and all that, but at least that one shot in this country has protected. Nobody's died of tetanus if they've had an initial tetanus thing there, but you can imagine that's not a fun way to go. So once we have that there, another enzyme, ATPase, everybody knows what ATP is? It's the energy currency in the cell. ATPase, ASE, means it's going to break that ATP apart, and it's going to release the energy to make the myosin head move. So you have that sitting on the myosin head, ATP comes in, it sits on that enzyme breaks that apart, and then you have the ADP and the energy needed to move. So if we're going to relax the cell, what things have to happen? We have to basically run everything in reverse. So we need to end the impulses coming in down there. We have the acetylcholine being removed from that space. We lose the stimulation. We can think about the T tubules as continually trying to scavenge that calcium, always trying to pull it back into side the T tubules. But I tend to think about it, I don't know if this is the right way to think about it, but if that impulse comes down, it basically, if I'm holding all my calcium, that impulse comes down, hits me in the back of the head, I drop all my calcium. Now I got to pick it all up again. And if I keep getting hit in the back of the head, I'm constantly dropping the calcium. That spreads into the myofibrils, but my job is to collect those calciums as best I can. So once we get the calcium back into the T tubules, we cover up the binding sites, the sarcomere relaxes, and everything slides back into its pre-contracted state. So what are the details? So that's kind of a macro level. What happens when we get down inside of the myofibril? In order for contraction to occur, we need a cross bridge. The myosin has to connect with the actin. The thick filament has to connect with the thin filament. Once that connection occurs, we also need a release of energy using the ATPase enzyme to allow that motion to occur. So what calcium does is allow that connection to occur. It allows the cross link or the cross bridge. So what calcium does comes in and it's going to take that tropomyosin, that troponin, it's going to pry it out of the way and open up the binding sites so the myosin head can reach across. So you can think of calcium as a crowbar that's going to basically open up or expose the binding sites. So once you have that happen, a sequence of events now start where you can have that muscle contraction. So if we're looking at the players in muscle contraction, you've got the thick filaments, thin filaments, these all kind of working together. You have calcium coming in and starting as a catalyst. You have energy down here, energy to move, and you have magnesium playing a part in catalyzing the whole reaction. So I used to have, okay, so at a molecular level, we start with the myosin complex flexed. ATP is attached to the side of it. Calcium comes in and binds troponin. That little pill thing we saw on the side, and it's going to pry open that binding site. So as calcium rolls the troponin out of the way, troponin is connected to that long black line. It's going to pull that long black line, and underneath that long black line are all the binding sites where myosin can attach to that actin helix. So calcium comes in, pry bars that open, exposes those binding sites. You have the myosin head, it's going to reach out as it relaxes to a binding site on that actin helix. Magnesium comes in, triggers the release of energy. Now the myosin head has energy to move, it flexes. Calcium is released and we start all over again. So that head is going to reach out, attach, flex. So if we start to pull,
and have clicked the link.
sorry.
Are composed of muscle fibers, which in turn are made of repetitive functional units called sarcomeres. Each sarcomere contains many parallel overlapping thin actin and thick myosin filaments. The muscle contracts when these filaments slide past each other, resulting in a shortening of the sarcomere and thus the muscle. This is known as the sliding filament theory. Cross-bridge cycling forms the molecular basis for this sliding movement. Muscle contraction is initiated when muscle fibers are stimulated by a nerve impulse and calcium ions are released. The troponin units on the actin myofilaments are bound by calcium ions. The binding displaces tropomyosin along the myofilaments, which in turn exposes the myosin binding sites. At this stage, the head of each myosin unit is bound to an ADP and a phosphate molecule remaining from the previous muscular contraction. The myosin heads release these phosphates and bind to the actin myofilaments via the newly exposed myosin binding sites. The two myofilaments glide past one another, propelled by a head-first movement of the myosin units, powered by the chemical energy stored in their heads. As the units move, they release the ADP molecules bound to their heads. The gliding motion is halted when ATP molecules bind to the myosin heads, thus severing the bonds between myosin and actin. The ATP molecules are now decomposed into ADP and phosphate, with the energy released by this reaction stored in the myosin heads, ready to be used in the next cycle of movement. The myosin heads resume their starting positions along the actin myofilament and can now begin a new sequence of actin binding. The presence of further calcium ions will trigger a new cycle.
So if you were going to do this in simple words, how would you describe this from happening? Describe this as happening. What happens first? Probably from your perspective, we say calcium comes in and does what? Binds, then does what? It's going to pry open and expose those binding sites. Now that there's a binding site, you can have what? Binding. Myosin can reach across. And then we have the ratcheting effect of myosin climbing up. If it's got 6 actins around it, it's got 6 different places it can attach. I assume they're asynchronous, and it basically climbs up those actin molecules. What happens when we take calcium away? Well, that's certainly one part of it, but what did we take the calcium away? What happens? T-tubules are picking up the calcium, taking it back, putting it back in there for storage. We take it out of the myofibril, out of the sarcomere. What happens? We don't have the crowbar anymore. Everything snaps back into place. We don't have the opportunity to link anymore. So that myosin's been climbing up the actin helix. If there's no place to attach anymore, what happens? Slide back to where we started, right? So if somebody takes the rungs off your ladder, it's kind of hard to stay on the ladder. So calcium comes in, opens up the binding site. You have myosin reaching out to those sites using energy. It's going to climb up those sites in a repeated pattern. We assume that there's asynchronous, so at least some of the myosin around that cylinder of myosin complex are always attached. So they're not all reaching out at the same time. So if we're looking at our system here, myosin is going to basically work its way up these things there as we have the slide past each other. We're going to bring the Z lines together. And that whole unit's going to contract. We take the calcium out. It's going to relax and go back to where it started, assuming there's tension on the muscle. So different thing there. So if we're looking at it, what's staying the same and what's changing? So we had a discussion and there was an argument in the A and P. They're on their past muscles right now. But they didn't have to talk about milk fever and things like that. What's moving relative to the other? And the demo we did in class, which we'll do here in a minute, they're well, the myosin doesn't move, the actin moves. Theory, the actin, the Z lines are coming together, they're moving all there, whereas this is anchored in the middle, so it's gonna climb up. I said, she's like, well, the actin moves, not the myosin. I said, it depends on where you're standing. If I'm standing on the actin, the myosin is gonna look like it's moving past me. If I'm standing on the myosin, it's going to look like the actin is moving past me. So both are moving past each other. There's a certain amount of movement there. So if we're thinking about hypocalcemia in our transition cow stopping muscle contraction, how does it stop it? Does it A, stop the nerve impulses to the myoneural junction? B, does it stop the stimulation of the sarcoplasmic reticulum to release calcium? C, stopping the movement of troponin complex to open up the binding sites or stop the ratcheting action of the myosin complex. When a cow has milk fever, what's going wrong in that sequence? Why does she go down? Why can't she stand up anymore? We've got no calcium. What's the problem? We're looking at C is what doesn't happen anymore. There's still stimulation to the sarcoplasmic reticulum or T tubules to release the calcium, but there's no calcium there to be released. So it's still getting stimulation. What happens is we're not prying open those binding sites anymore. We can't contract anymore. So that's the problem when we're thinking about milk fever, hypocalcemia, what's happening. So here's our sequence. You guys ready to try it? So who would like to be calcium? Everybody's going to participate in one way or another. So you can be myosin, you can be actin, or you can be calcium. Who would like to be calcium? Okay, so if you could stand by the door. Everybody else, I need 8 people on one side, 2 people on the, well, I guess we're 7 and 2. We're missing a student today. 7 on one side, be the actin helix, myosin. We need 2 people to be our myosin heads. Who wants to be myosin? Who wants to be myosin? 1, 2. Everybody else on this side, over by the tables. So over here, we're going to be the actin helix. We're going to stand like this with our hands behind our back. So myosin, we'll stand one here, one there. So how does a signal work? How does muscle contract? We've got the actin helix here. We've got the myosin heads here. We've got calcium over there. So what has to happen? Muscle contraction. So calcium is going to come in. What is that going to do for us? Open the binding sites. So your hands, actin helix, your hands, you now have a binding site. What can we do with that binding site? We can reach across with our myosin and connect. Okay, so we're connected. You gotta keep those other sites open. So as long as we have energy, what's that myosin complex gonna do? It's gonna reach out to the next one, so let's go this way. So reach out to the next binding site. Energy is released. What do? What does myosin do? Flex, so that means you're stepping to the right. Make sense? Then we reach out to the next binding site. Energy is released. We step one more time and we continue to slide along that. What happens if calcium goes away? Calcium goes back out into the hall. First thing that happens is the binding sites go away, right? We've covered them up. And then what happens? They go back to where they started. So it's just helping you visualize what we're doing here. Okay, so calcium comes back in. Nerve impulse releases that calcium for the T tubules. Calcium is here. Binding sites are open. We cross bridge across the way, wherever you want to start. So we're going to start there. We're going to reach out to our next spot. ATP comes in, energizes that head. It flexes. We move over. It keeps going all the way down. As long as calcium is present, it will keep reaching across and flexing. Calcium goes away. So the muscle can relax and we might slide back. If there's tension on that muscle, we'll slide back to the original position. If there's no tension, if I left my arm here, it might stay where it is, but it now has no connection and can go anywhere it wants. Is that helpful to visualize what we're doing? You guys ready to do it without my coaching? Okay. Carrie Ann, you're going to start. What's happening? Narrate it. So the energy is used to flex, that climb. So we did this from the perspective of actin. So what might also be what happens if you go back to calcium leaves, come back to a start, slide back to your beginning. What happens if the actin moves and the myosin doesn't? So calcium comes in. We opened up our binding sites. We pry that tropomyosin troponin out. So what's going to happen? Actin is going to cross-link. You're going to reach out to your next spot, right? Now what's going to happen? Actin is going to step to the left. It's going to pull as it flexes. It's going to pull or to the right, the other left. So you guys are going to step to the right as that flexes. It's going to drag you by. It's going to ratchet you by. So we reach out. We flex again. We move one step to the right. We reach out again. We move one step to the right. Calcium goes away. Everybody slides back to where they started. So depending on where your perspective is, it depends on what's moving. It's all relative. Do you guys want this up on YouTube so you can refer back to it later? Okay, what would you like to do? Would you like actin to slide or myosin to slide? Okay, so it happens. Okay, can we make this work? So,
25.
okay, so we start the muscle nerve impulse. So what happens? Comes in, so you're gonna have to come in just a little bit farther, right about there. Okay, so back up. I think I got it ready to go. So back up. Let's go back to calcium coming in. In 3, 2, 1. Calcium comes in. Binding sites open. We have cross linkage, cross binding, cross bridge. We reach out. Energy is released. We flex. And myosin is moving. Uses up that energy, reaches out again. Continuing to flex and move and slide past the actin helix. Calcium leaves. Cross bridge is gone. Binding sites are gone. We slide back to the beginning. There you go. Did everything you hoped it would be. Okay, why don't you take your break now? We'll start again at 14 of. Okay, so any questions about muscles? So let's go to another muscle, our heart. So we're going to talk about the bovine cardiovascular system. We'll start with the heart first, and then work our way to blood vessels and blood. So the goals of this section, I want you to be able to do the red blood cell sequence. If you're a red blood cell, can you put all the parts in order? Where do I go first? If I start in the right ventricle, what do I go next? What things do I pass? How do we go through the system? And then when we get to the heart function, I want you to be able to put the heart events, heart sequence in order, the events that happen. So those are our goals for this section. So we'll start with kind of the purpose. So what do we know about the cardiovascular system? Pumps blood. Everybody knows about that one. So with that pumping of the blood, we deliver oxygen and we remove carbon dioxide. So that gas exchange occurs in the lungs. We deliver nutrients and remove wastes. Those exchanges occur at the tissues. We move stuff around from different parts of the body to other parts of the body. So that's the highway. That fat will get moved from, say, the liver to the muscles or vice versa. Adipose tissue to the muscle, wherever it needs to go. And our cardiovascular system is very intimately involved in thermal regulation, helping cool us down in the summer, helping keep that heat back in the body in the winter. So we tend to have more blood flow to our skin in the summer, less in the winter. So if you're looking at the vertebrate cardiovascular system, your heart has 4 chambers. Anybody here done the frog? Dissection somewhere along the way? How many chambers does their heart have? 3, right? 2 atria and 1 common ventricle. It's not the most efficient system, but they tend to be a little bit smaller and they can get away with that. They're also cold-blooded. They don't need as much oxygen to survive. So 4 chambers, we got 2 rights, 2 lefts. We've got 2 atria, 2 atriums, 2 ventricles. You guys had this before, somewhere along the way. So there's 2 distinct and separate circulatory systems in the body. Most of you have probably talked about the one that services the body. We pump out through the aorta, we service the body, but maybe we don't talk as much about the lung or pulmonary system. So you have the lung or pulmonary system, and you have the body or systemic system. We look at the lung or pulmonary system. It's characterized by low volume, short distance, and low pressure. So we're going from the heart to the lungs and back. It doesn't require a lot of pressure differences to get that done. It's all inside the thoracic cavity, so that gets done very quickly. We have a short distance to cover. If we think about the capillary bed that forms in the lungs, we have something that's very interconnected. It's almost a net or a mesh that we're looking at. The blood is going to flow through. We think about sheets over all the lungs, a sheet of blood. We're spreading things out. We're thinning things out. We're increasing the surface area on that liquid. So you're looking at probably half a football field in your lungs, spreading that blood out over basically 1 red blood cell thick over a very large area. In the lungs, oxygen is added and carbon dioxide is removed. And we'll get more into that as we move forward. So the other circulatory system or circuit that we're going to look at is the body or systemic. You've got high volume. That's where most of your blood is. It's the longer distance of the two, just from the heart to the feet and back. And because we're covering a high volume, covering long distances, we need a higher pressure system to make that system work. So it's going to handle much higher and thicker system. The capillaries are not as highly interconnected. You don't have a mesh of capillaries as you get into the body. But as a general rule, every cell in the body should be near or close to a capillary. And we've all about almost. We saw in the bone that there's some distance between the two, between the osteocyte and the Haversian canal. And there are some spots there where maybe we're 2 or 3 cells away, but we need some capillary nearby to provide gas exchange, to provide nutrient exchange. So the body is going to take out that oxygen to use. It's going to take out nutrients to use. And then as its waste products, it's going to put carbon dioxide back into that capillary bed. It's going to dump its waste in there to be taken away and be processed wherever seems most appropriate. So 2 distinct circulatory systems exist. When we're thinking about the cardiovascular system, we generally go from artery to capillary bed to vein. And we're thinking about arteries, we're not thinking about oxygen content. The one we kind of exposed to, the aorta, the body system, yes, the arteries have oxygen in them. But in the pulmonary system, there's no oxygen in the arteries that leave the heart. So it's more about the direction of flow in relationships to the heart. Generally, arteries are going to leave the heart. Veins are going to come back to the heart. And that's not 100% perfect, but that's a good way to think about it. So we're not thinking about gas or oxygen content. We're thinking about the direction of flow relative to the heart. So artery takes blood away from the heart. Capillaries are going to connect the artery to the vein, and then the veins are going to bring the blood back to the heart. What connects the veins to the arteries? The heart. So we have veins coming in one side of the heart, arteries going out the other. So this is a nice diagram here that kind of goes through all the vessels that you're dealing with. So you start out at the heart, we come back from the body, we go out the pulmonary trunk, pulmonary arteries, through the capillary beds, we do the gas exchange there. As a general rule, blue is shown as deoxygenated blood. Red is shown as oxygenated blood. Is anybody ever here drawn tail blood? So what do you usually see as colors there? If you get the vein, what do you get? Is it blue? No, it's a dark maroon. If you go through the vein and hit the artery, which is deep in there, you know it very quickly, right? Have you ever done that? You go in deep enough, you will hit that artery, and all of a sudden you got high pressure coming into that vacutainer, and it's bright red. Oxygenated blood is very much bright red, whereas our deoxygenated tends to be more maroonish, brownish, not the blue. But that's the convention, that the blue is deoxygenated, the red is oxygenated. So once we go through the capillaries, we do our gas exchange, we have oxygenated blood returning to the heart. It's going to come in the left side of the heart. It's going to be pumped out through the aorta, the arteries, the systemic or body capillaries. We work our way back to veins, eventually get to the vena cava, and we get back to the right side of the heart. So as we said, generally represents red and blue, oxygen poor, oxygen rich blood. So some general rules if we're thinking about the pathway. We've already said that arteries leave the heart, veins return to the heart. The atrium is always before the ventricles. Right side feeds the lungs, left side feeds the body. And we're talking about the left of the animal, or your left, and the or right, depending on where we are. Arteries move by pressure. Veins move because of valves that keep things flowing in one direction. So we've got some diagrams here you can practice on. If I spot you the right atrium right here, the blue there, you should be able to go through in the pattern, working your way through. These squares here, you can say whether it's oxygenated or not oxygenated if you want to practice that way. So we also have another blank diagram here we'll go through. So this is that diagram here. We start in the right atrium. We're going to go through the AV valves. The AV valves are going to be the valves between the atria and the ventricle. So right atria, AV valves, right ventricle. We're going to go through the semilunar valves and be pumped out to the right and left lung. So you've got the common pulmonary trunk that's going to split into a right and left pulmonary artery. That's going to go to a capillary bed. We're going to return oxygenated through the pulmonary veins on either side here. They're going to come in. We go to the left atria through the AV valves on the left side, the left ventricle. We pump out through the semilunar valves into the aorta. We go to the body. We go through the capillary beds, and then we come back through the back half of our cow, comes back through the inferior vena cava. The head and front leg come back in the superior vena cava to the right ventricle. So I assume you've had most of that before. Is that a fair assessment or not? Who has not had that before? Okay, so you probably focused on a little bit 30,000 right and left. So how do I know on my diagram which one's right and which one's left? So there are some clues in the drawings to look for. So what did we say about the body circulation associated with the left? It's oxygenated and high pressure. So the ventricles are going to have to generate more force. So in the diagrams, they're generally thicker. And if we get to our dissection, you'll see that those walls are thicker as well. We also know it's oxygenated, so we see that red color. So the convention is that if you're looking at a heart on a page, you're looking at somebody standing in front of it and looking at their heart. So you're looking ventrally or from the front of them, not from the back. So it's opposite of what your right and left is as you sit there staring at it. So this is from the Atlas of Bovine Anatomy. So which one's which? Which side of the heart is which? Left side, because it's thicker and we've got more oxygenated, represented by the white arrows. So we flipped it from the previous diagram here. So coming in, atria, ventricle, out to the lungs, coming back on either side, coming back here, we pump out through the aorta and through the rest of the body. So if we're looking at our heart, we generally think about what we think about when we think about a heart is mostly the ventricles. When we get that heart out of our dissection, you're going to see that the atria up here are little floppy things. Their job is to contract, but they don't have to generate a lot of force. Whereas our ventricles have to perfuse some circuits, and in doing that are going to have to pump out a lot more strength. So they tend to be very thick, and they tend to be, if you put them on a table or whatever, they're going to hold their shape. Whereas the atria up top, we got a lot of floppiness happening, and they really don't hold their shape. So looking at it, sagittal section, what are we doing? Cutting through the middle of the heart, sagittal. So again, left side, right side. So we've taken this heart and opened it up. Has anybody here heard of you're pulling on the heartstrings, my heartstrings or anything like that? So has anybody ever had heart or opened up or dissected a heart and seen those heartstrings? The little tendons that are going to hold the valves in place, preventing them from flowing backwards. So chordae tendinosus. So you've got the AV valve here. You've got the things there that are going to prevent that valve from blowing backwards when it's under pressure. So the flow of blood is this way. So we have those strings on the other side to prevent that from going backwards. So you can see that valve replacement in a heart is not as simple as it might sound. Although when I was a kid, there was a woman in our church that had one of the very early valve replacements, and it was just a check valve that they put in. And you could take her pulse from across the room. She was always clicking as that ball went up and down in the, everybody seen a check valve? You have the circle here that the ball sits in, and then you got the cage that there. So anytime the heart would pump, that ball would hit that circle, that plastic. And if you didn't know, if you were new there or hadn't been around a lot, she loved to be the person in charge of collection, because that's when everything was very quiet. And she'd go around and they, people were just like, what's that clicking sound? And that was her. So she had one of the very early things there. So it's a very simple system that allows things to happen. Now they put bovine and pig valves in you and things like that. There's probably more organic and probably fits a little better over the long haul. So we have our blood system going around the outside. You're going to have early branches off the aorta are going to provide blood to the heart, and we need all this tissue to be perfused. One of the things that a heart attack can lead to is if we starve this tissue of oxygen, that can lead to a heart attack. And I have a friend that's he just had his 4th shunt put in. He comes from a family where everybody's high cholesterol. So keeping these open is what's going to keep you alive for the long run. You have those diagrams there. Again, you've got left side of the heart, right side of the heart, atria, ventricle. We leave to the pulmonary trunk going up that way. We come back into the left ventricle here and here, pulmonary veins, atria, AV valve, heartstrings, holding that AV valve. We get pumped out through the semilunar valves into the aorta up top. So chordae, tendineae are these tendons that are going to prevent that valve from going backwards. So if you don't like blood, you might want to close your eyes for a moment. So these are the dissections we've done up at the dairy. So this is a cow's heart. So let's see, looking at something about that big. Got a calf's heart, probably a little bit bigger than fist size. So this is about the size of your heart. I'm amazed at that, how big that is, but when you're talking about perfusing something that's close to a ton. So you can see that calf heart. You got the little floppy atria up here, one ventricle on this side, one ventricle on this side. If I had to guess, this is left, but I don't know for sure in the picture. So you've got the vesicles coming out. This is probably the aorta coming out. The other one's probably the pulmonary artery. So which is which? Left ventricle, much thicker wall here. Thinner wall over here, that's the right ventricle. And you can see those a little bit better. You got the flap here, that's the valve, and then the tendons that are anchoring that, preventing that when we get that pressure. When the ventricle contracts, making sure the AV valve doesn't blow backwards. We want all our valves to be one way. So again, that calf heart, looking at how things are. We got a cow heart pulled out of the freezer. So again, much thicker wall over here. We've got all the pieces associated with holding that valve in place. We've laid open this valve, the AV valve. So the sinuses for the atria are right here. Then we're going down into the ventricle as we move forward. So if we're thinking about a simple 2 circuits, we come in on the right side, deoxygenated blood. We pump out from the right side of the heart to the lung capillaries. We're going to do that transition where we dump the carbon dioxide. We pick up the oxygen. That's going to go to the left side of the heart. That left side is going to pump out to the entire circuit of the body. So that's a simple kind of illustration of the 2 circuits. This is more closer to accurate, but again, we've got simplifications that are happening. So we come in, I think you have this list there somewhere. So the numbers on this page match the numbers on this page. So if we start with the 1, we have deoxygenated blood in the right ventricle. We're going to go out through the semilunar valves into the common trunk. And then we're going to split left and right pulmonary arteries. We go to the 2 capillary beds in the lungs where we make that exchange. The oxygenated blood is leaving, coming back through the pulmonary veins. We go back to the left side of the heart, our left atrium. Left atrium right here. We've got the AV valves that we move through. We've got the left ventricle right here. We pump out, we get enough pressure. These valves, the semilunar valves open. We leave through the aorta. That's going to split into ascending and descending parts. So on us, it's going to go up to the head or down to our feet, or a cow, it's going to go anterior, posterior, craniocaudal. Perfuse the upper part of the body, perfuse the lower part of the body. That oxygen is removed, CO2 is put back. In both of those areas, we come back either from the inferior, the lower half of the body, the inferior vena cava, or the front leg or the head, we come back to superior. We've got two vessels emptying into the right atria. That's going to push blood through the AV valves back to the right ventricle. So those are the things we're looking for with the valves. Can you put them in order? So give some thought as to how you'd like to be tested on that. So this should walk you through that set. Again, we're following all the rules we've established so far. So any thoughts on how you're going to learn all this? One way to remember, and you'll appreciate this as you get older, is song. Why do we remember songs? Why do advertising have jingles? Because songs tend to stick in our brain. So this was found by a past TA for this class. Let's see, it's an old time TV show, but
Field capillaries tube in the oxygen, blood of the cells, tissues in the cells with operations, CO2, which is carried to the kidneys and the veins. To back down to the atria of the lungs, and where we started in the heart.
so sometimes songs are a good way to do it. You certainly got all the parts that we talked about in there. What was the difference that you heard? Bicuspid valve and tricuspid valves. So those are the ones with the heartstrings on them. We're not going to distinguish between the two. We just call them the AV valves. So why do we call them the AV valves? Atrioventricular valves. Because that's what they sit between, right? They're the connection between the atria and the ventricles. So any questions on the path or the parts of the cell or bovine circulatory system? So I've got a couple of more study aids. I'll bring them to next class. We'll give some thought. So heart function. So we've got all these parts working together to deliver blood to your rest of your body and your lungs. How does it all work together? So the best way to go about this is to think about it, at least for me. The first thing that happens is electronic or electrical. Those electrical events lead to muscle contraction. And then muscle contraction leads to a pressure change that basically puts everything moving forward. I know there's some diesel boys that have been in this class. Oh, it's just like hydraulics. Yeah, it's the same thing. Everything's got one-way valves, and you increase pressure here. Everything moves from a high to a low. We move through the check valves, and that's why we go only in one direction. So electric leads to contraction, leads to pressure events that have everything that's there. So we'll start with the electrical. Well, we got to talk about getting things. So cardio contractions happen in a coordinated manner to move blood through the body. We have to think about cardiac output. So we have how much is ejected on each beat. Milliliters is what we usually think about for humans. I would imagine you could go with a larger amount for cows, especially a full-grown cow. And then the number of contractions or number of beats per minute. So if you take the milliliters per contraction and then contractions per minute, you cancel out contraction, and cardiac output is milliliters per minute or volume per minute. So how would we modulate or adjust our cardiac output? We either have to change the amount per contraction, increase it or decrease it, or change the number of contractions that we have per minute. If we're looking at our cardiac output, we're going to rely heavily on our pacemaker cell in the heart. The first thing that's required or needs to happen is the pacemaker in the heart sets the rhythm, sets the number of beats per minute. It's called the sinoatrial node, or we'll just call it the SA node. It's going to sit on the right atrium, and it's going to set the pace. Now that SA node, the sinoatrial node, can take input from the nervous system. It can be increased, told to increase its rate of sending out pulses, or decreased depending on nervous system input. Everybody's had that near car accident, right? The nervous system sends a signal to your atrium, your SA node, and says, start picking up the pace because something bad's happening. We need more blood. We need more cardiac output to get things done. And you sit there in the car, and how does it feel? Anybody had that? You have a near miss in a car, and you get that flushed feeling, and your heart starts racing. Because normally when we're attacked, we'd have to run somewhere, fight or flight. So the body is getting ready for that. So we're thinking about cardiac contractions. We're going to have to think about the electrical system as based off of two nodes. So the first we've already mentioned, the sinoatrial node, the SA node, or the pacemaker. It sits on the right side of the heart, on top of the atrium. It's a specialized region of cardiac muscle that sets a rhythm for our heart contractions. Goes up to a threshold, fires, and we have a contraction. Threshold, fires, contraction. And this can sort of do things on its own, more or less. Has anybody worked in any sort of slaughter facility? So I put myself through college on a game bird farm, and every Wednesday we'd process birds for the gourmet food trade, which means we put them in crates, we brought them in, we basically cut off their heads, we let them bleed out, and then we put them into a scalder. So everybody dealt with a scalder or not? So 142 degrees to loosen up the feathers. Then we put them through a plucking machine and takes all or most of the feathers off. And then given the timer of that, they're spit out onto a stainless steel table, and we chilled them down from there. Every once in a while, you'd have a bird or a duck that had been basically bled out put through the minute and a half in the scalder, 30 seconds in the plucker, and it comes out on that table, and you pick it up and its heart's still beating. It's got no head, it's got no feathers, it's been abused, but every once in a while, that signal nose, SA node hasn't figured out to stop yet. So it's still pacing itself, still going through. It works independently unless it gets outside suggestions from the central nervous system. The other one is a atrial ventricle node, or the AV node. So where do you think this one is? So it's going to sit there in the center of the heart. You got two atria, two ventricles. It's going to sit there in the middle. This is going to be, has anybody here done brake work or car or clutch work with hydraulic clutches? So this is the master cylinder. If we're thinking about brakes, this is the slave cylinder. This AV node does whatever the sinoatrial node tells it to do. So it's going to follow, it's going to do whatever needs to be done associated with that node. So everything starts with the pacemaker, the SA node, and then the AV node follows suit. So if you're looking at how that kind of works out, you have the SA node sitting up on the right atria. That's going to fire. It's going to spread a signal out across the top of the heart. As that signal goes around, it tells those atria muscle cells to contract. So as that globe or kind of hat of electrical signal there spreads out, it's going to contract those atria. They're going to push blood to the ventricles. As that happens, eventually you're going to hit the AV node, and that's going to fire. So you've got the atria contracting first. As that finishes up, it hits the AV node. That sends a signal to the bottom of the heart, and we contract from the bottom up. So the electrical signal flows back up to the atria. So why does it have to happen that way? Why does the signal just keep going down? Now the exits to the heart are up here, so we need to squeeze from the bottom. If we continue to squeeze down, we blow out the bottom of the heart. So you need that jump of a signal. There's lots of names for these specialized nerve cells and connections. We contract from the bottom up. It's going to push out through the pulmonary artery and the aorta, moving through the body. If anybody watches TV shows with the heart beating, right? So each of these electrical events is associated with a spike in electricity. So you've got this part here is the atrial contraction. This part here is the ventral contraction. And then this is repolarizing, resetting for the next contraction or next electrical event. So has everybody got the electrical events down? So we said electrical leads to contraction. You can see where we're going with this. Leads to pressure events that are going to change the blood and move it to where it needs to. So we'll stop there today. I got you 5 minutes back. We got 55 to go.